What if your mood and immunity were partly shaped in your intestines? The gut is a control hub, not just a food tube. Its microbes signal through the two-way gut-brain axis using nerves, hormones, and immune messages. One route runs through the vagus nerve. Another involves the intestinal immune system, where a large share of immune cells are associated with the gut. In this scene, focus on three pathways: microbial metabolites, immune signaling, and nerve activity-linked digestion to brain function and body defense. Inside the intestinal lumen, the microbiome is a working ecosystem, not a single organism. It includes bacteria, viruses, fungi, and other microbes living mainly in the intestines. Their importance comes from metabolism. Food, drugs, stress signals, and infections change which microbes are active and what they release. Those outputs include small molecules the body can detect at the gut lining. The key idea is input to output. What enters the gut is processed by microbes into signals that can alter how the body responds. Microbes convert fiber into short-chain fatty acids such as acetate, propionate, and butyrate. These molecules strengthen gut lining cells and change local immune cell behavior. Other microbes modify amino acids into serotonin precursors and related neuroactive compounds. Gut cells and immune cells detect those signals and release immune cytokine signaling. The body carries this information through three major routes, including the vagus nerve, hormones, and circulation. Brain activity and immune tone can shift because the original message began as microbial metabolism. Short-chain fatty acid output depends directly on how much fermentable dietary fiber reaches the colon. With more fiber available, microbes generate three main products: acetate, propionate, and butyrate. Butyrate serves as a fuel source for colon lining cells. That energy supports mucus production and tighter junctions between neighboring cells. A stronger barrier limits contact between immune cells and stray microbial fragments. In low-fiber conditions, fewer fermentation products are made, and immune activation becomes easier to trigger. The hardest part of microbiome research is separating cause from constant change. 6 common factors alter it at once: diet, antibiotics, infections, stress, sleep disruption, and aging. That means a microbial pattern seen with low mood or immune dysfunction may be a consequence, a contributor, or both. Measurement adds another limit. A sample can show which microbes are present, yet miss what chemicals they are producing that day. Researchers often find associations first, while direct causation takes controlled studies and repeated measurements. The evidence comes from 3 main categories, and each answers a different scientific question. Animal microbiome experiments provide causal leverage because microbes can be changed under controlled conditions. Those studies show shifts in stress responses, immune signaling, and intestinal barrier measures after microbiome transfer or depletion. Human cohort studies add realism by linking stool sequencing patterns with mood, cognition, and inflammatory markers across large groups. Diet intervention studies and antibiotic perturbation studies show communication pathways are real, while one microbe, one disease claims remain less certain. The microbiome is not fixed. It shifts across 6 major drivers: dietary fiber, antibiotics, acute infection, psychological stress, sleep disruption, and aging. Some changes appear within days, while others accumulate over years. Antibiotics can reduce diversity and alter metabolic output. Acute infection can temporarily reorder which microbes dominate. Stress and poor sleep also act in the opposite direction, because gut-brain communication is two-way. That constant movement helps explain why the same person can show different gut-linked brain and immune patterns over time. Gut signals reach the brain through several channels at once, not through a single line. The vagus nerve carries sensory information from the intestine toward brain regions that regulate autonomic state. Hormones released by gut cells add a second route and reflect nutrients, microbes, and stress. Immune messengers add a third route when intestinal conditions shift. Microbes also shape neuroactive chemistry by modifying serotonin precursors and GABA-related metabolites. The result is two-way signaling, with brain output changing gut function while gut conditions alter incoming signals. Microbiome research follows a 5-level evidence ladder, and each step changes how strongly a claim can be interpreted. Association studies can show that people with depression or inflammation carry different microbial patterns. Causation remains uncertain because interpretation risk is high. Medication, diet, illness, and lifestyle can all reshape the same samples. Longitudinal sampling tests whether microbial shifts appear before symptoms. Intervention trials, germ-free animal models, and fecal microbiota transfer add stronger directional evidence. Human certainty remains mixed for many specific disorders. The intestinal barrier works like a selective filter between gut contents and underlying immune cells. Short-chain fatty acids, especially butyrate, help intestinal barrier cells stay nourished and closely sealed. When that barrier is disrupted, more microbial products reach immune cells and shift local signaling toward inflammation. Because a large share of immune activity is linked to gut tissues, that shift can spread beyond the intestine. Signals generated there can alter overall immune tone and send inflammatory cues that also affect the brain and energy balance. Microbiome is adjustable across life, not a fixed personal signature. 6 major categories shape it: diet, antibiotics, infection, stress, sleep, and aging. Dietary fiber changes microbial fuel supply and shifts which groups expand. Antibiotics can sharply reduce diversity and alter community output, with recovery varying by exposure. Stress and poor sleep also change gut conditions that microbes experience each day. That means ordinary routines can recalibrate microbial activity, which changes the signals available to the brain and immune system. The clearest conclusion is modest and strong. Gut microbes participate in signaling that can influence mood, cognition, and inflammation. That general communication is supported across human studies and animal models. The weaker claim is far more specific. A microbiome association with depression, cognitive change, or immune disease does not prove direct causation. Microbial patterns also differ widely across people, so no single profile explains every outcome. The useful model is a network, not a single bug or simple diagnosis. Diet, environment, host biology, and time all shape the signals being sent. Science supports participation. It does not support one universal microbial script for every brain or immune state. Thanks for watching. If you enjoy clear science explained with strong visuals and evidence, subscribe for more long-form science videos.